So a little bit of review on earlier class. Till the last class, we have kind of covered big data bioinformatics. We have covered, genomics, and we have covered, thereafter moved on to transcriptomics. And we learned what is RNA-seq data, what are the processes of analyzing RNA-seq data, and that aspect, how can you use the server and run the pipelines. We have a lot of demo pipelines out there. The reason being that to make your life simple, about not to think about what kind of exact input or output need to be given, so that you can get an understanding of the input and the output that needs to be given. And when it comes to getting the projects at the end of the course, the last month of the course, you will have you'll have ease in running the pipelines, and would be able to do the analysis all by yourself and get the interpretation done out of that. So as a part of that, you can see a link on your screen right now, which is a tiny URL link. I think most of you or all of you have the presentation with yourself. Now, please go to the link. You will find a feedback form out there. Whatever we have covered in the transcriptomics has to be, we want to understand what is your understanding about the course till we have achieved, till we have come to till date. And please remember that the certificate of participation is also associated with your feedback, because we are a kind of organization, although we are a company, we always stress on the fact of the quality of the stuff. So we need to understand what quality we have been really able to cater to you as a part of this course. So that's why we call it extended MOOC, extended massively online course, a strong feedback system. And we would like to learn from you that what have you really learned? What did you, what kind of knowledge you didn't have, and what kind of knowledge you have finally come up to by doing our course. That will be your evaluation too. You can yourself evaluate your knowledge, and at the same time, you'll get a pointer that what kind of things need to be done to enhance your knowledge. I think, our courses are sufficient, is perfectly all right for giving all those essential pointers to embark into the domain of data science as biologists. So if you have registered for this course, and I think you're having a good experience, so that's what it is. We would like to know your experience about the course through this feedback form. So fill it up. Many of you have filled up, but those of you who haven't, I would request you to fill it up, and then it would help us to understand. So the last week we did on transcriptomics. I think as a part of that, transcriptomics 1 and 2 from the educational pipeline has to be completed. So edu.t-bio.info/courses will take you to that particular page where a number of courses are there: Introduction to Bioinformatics, Metagenomics, Transcriptomics, and genomics as well, and also machine learning. So it is wise and it is advisable. It is requested. I would insist. I mean, all those languages really mean a lot. And go to those educational courses, complete them. It will give you that elaborate understanding of the topics that we are discussing. Progress till date. This is the progress since September 10th, when we had our last class. We can see many of you are using the server as well. Today is September 17th. Many of you, I think, have started doing the course, or those of you have not started. So the leaders of of this week, I'm sorry, the slide might be a little hazy. So Debanita has been doing pretty well, using a lot of educational materials and server. Shubhrajit happens to be one of our research interns. He's actively involved in running many pipelines connected to a project that probably one of you would also like to do, devising your own project. So Shubhrajit has been asked to do a project by us because he's a research intern. We have opportunities to take research interns. Shubhrajit has done well. That's why you have inculcated him into the team. And you also have a similar kind of opportunity if you really can show your skill set and really can own your skill set and earn it at the end of this course. Devalina, good. I mean, I have seen that many of the pipelines that you have run, and also the educational courses you have been doing. Ruchika, Dahiya, and Sunita is also there. Sunita has been running a lot of education. But what about the rest? I mean, I can see significant zeros in the education, but how can you just run a server without having the data set on your own? Velu, Shomalo, Kulbhushan, Swati, Jeffrey, and Aravind, and Somadeep, Swaranjit Prasad Pal, Srijani, all these people. I would really like to know that what data, where did you get the data from? How do you get the data from without doing the educational courses? Just took the data from the educational platform? That's not wise. I mean, that's really not wise. So you have to understand what those data sets mean. Now, if you have run the server, I would really like to get back to you, each of you, to understand what sort of analysis you have done and what sort of interpretation do you really produce, which is the result of the server. So the way we might do it similar way in the next workshop, which is coming up, like to share our screen. I think many of that sharing screen kind of helped you to understand. I can see some chats. Okay, so not very significant. So here, so that's why I would really like to talk to you, all these people who have kind of 0 marks in the educational. And be remembered that your points also matter. That will really make you feel or make you understand at what level of understanding you are. So all these composite things is important. So today is the day when we start with something called metagenomics. Now, I do not know whether how many of you are really aware of this word metagenomics. I think all of you have been pretty aware of the concepts like genomics, transcriptomics, proteomics. People have been talking to us a lot that, don't you have a course in proteomics? Don't you have a course in metabolomics? Yes, of course. We have all those necessary pipelines by which you can run metagenomics and proteomics data sets. But yes, we are coming up with all those specific courses on multi-omics perspective. The remaining are basically metabolomics and proteomics as a whole. And there are a lot of other omics data like phenomics and all these things. So these are all things which are going to come up step by step, but this is only the first step of the kind of learning that we are trying to give you. So metagenomics deals with microbiomes. So why microbiome is so important? Now, I would come to that point a little later. So metagenomics encompasses all those important aspects of genomics, proteomics, and transcriptomics, focusing on a particular organism. Now, what is this organism? These organisms are microbiomes. So can you tell me why microbiomes are so important and why it is so important for studying microbiomes? Can you unmute yourself and tell me, or you can write in the chat, whatsoever is comfortable for you. We have some answers. Okay. Okay, so okay, but I think Modi, you had an access to the educational platform long way back during the start of the course. So I would really like to know what kind of what educational courses you have really done and earned certificate. Okay, Velu, I think use the dataset link which we provided in the class for running pipeline. Yeah, I also said that the dataset that has been shared with you is connected to the course in the educational platform. So did you do the course? If you have not done the course, I would urge you to go back to the course. So Shomudeep, being busy in exams, I think, this is the professional class. So that's why being busy or not being busy is something else, which is your personal matter. I don't really would be like to discuss about that. But, I mean, I don't know. I mean, you have paid for the course, so you should be doing by your own. So we are not trying to teach you ethics or morality here. We are trying to give you all those professional, capabilities and upskilling, which is required for your research, which is required for to get employable. So, and your university has really, put up this program as a part of the employability enhancement. So we are very clear about it. I guess at participants, we should also be very clear about it. So yeah, so what is metagenome? if I can ask you? I would, maybe all of you can see my chat. So responsible for health, source of novel pathways, and therefore products influence plant health as a consortium. Doesn't really mean anything. I've got so busy. Okay, thank you, Prakash. So don't worry. So what is microbiome? Velu has given a definition, or let's say a hypothesis, which is close, but pretty far away from the concept that I'm going to, what is metagenome is. What is microbiome and metagenome? Yeah.
Real quickly.
Somebody speaking?
Yeah, can you hear me fine?
Yeah, again, tell me.
Yeah, so I think about it, I study virology, and
Excellent.
when we think about quasi-species, where each virus in the population may have a different genetic mutation, if we were to just look at one specific virus, that wouldn't tell us what's going on in the population. But when we are able to sequence thousands or millions of the different viruses and look at all of the genetic differences, I think that's a good example of metagenomics.
Okay, I kind of agree, but also, I kind of agree to disagree. So in addition to the viruses, there is something called bacteria, which is kind of inhabiting your entire body. It is actually inhabiting many of most of the infectious disease that we have are kind of bacterial borne disease, and of course viral borne disease. But that's a separate, separate study. So that's vironomics, because the virus kind of have more closeness towards the eukaryotic. It shares both the eukaryotic and the prokaryotic characteristics. So that's how it is. So I would say that microbiomes are important because they cause infection, a lot of infection in the human body, a lot of infection to the population, to the animal population. If you remember the first slide that we did on introduction to bioinformatics, there's something which I showed about H1N1 virus, which is you are speaking about, but at the same time some plant pathogens and the organisms pathogens which inhabit, which really infect the organisms, the different type of domesticated animals like chickens, like horses, like cows, and all those things. So if you can really, if you really look at the pathogenic infection, bacterial population inhabit almost every corner of the ecological niche. And since we are a part of ecological niche, that's why, because we are also we are also infested with microbiome. If you remember clearly, that E. coli infection is a very bad type of infection which happens in our body. But E. coli do resides within our stomach. E. coli has that is there in our stomach. If you really look at so if you really look at your cecum, the cecum that is there in our intestine, cecum harbors a lot of bacteria. Our skin harbors a lot of bacteria. Our oral cavity has a lot of bacteria. So and they use humans as a host. So all bacteria, many of the bacteria use humans as a host, as a part of their life cycle, as a part of their reproductive life cycle. That's the natural biology of these microbiomes. So microbiomes are specifically those species which inhabit, which are unicellular animals. They're capable of reproducing very fast, and as a part of that reproducible cycle, they infect a lot of other organisms in the entire taxonomic classification of the entire ecological niche. And they can be beneficial. They are, I mean, if you remember that, there are so many probiotic drinks which are available. I mean, what are these probiotic drinks for? These probiotic drinks are basically, they are concentrated microbiome, spores, which are beneficial for our gut. So there are some brands across the globe. It can be, it can be probiotic yogurt. It can be some probiotic drink. Even curd has a lot of probiotics. That's why, people eat a lot of curd after serious infection. And nowadays, many of the antibiotics, or all these antibiotics are kind of, they have some probiotic element associated with that. After a series of infection, series of antibiotic course, doctors do prescribe probiotics because, as you know, the antibiotics kill randomly within our body. They get into our stomach, they can get into our system and kill randomly. They kill, they do not really classify between the good bacteria and the bad bacteria. So that's why microbiome is an organism which is there in our body for a very long time. And recently, the concepts are being developed that microbiomes play a significant role in the progression of the cancer. Microbiomes do play a significant role in deciding the type of person we are. So different type of biological concepts are coming over: epigenomes and microbiomes, metatranscriptomes, transcriptomes, all these things. So these are all kind of super controls within our body, playing inside our body, which has their own type of life cycles. So, and also metagenomes inhabit some of the extreme environments at the same time. So that's why we also call it extremophiles. They inhabit the sulfur vents. They inhabit deep sea oceans where the temperature is almost minus, let's say, minus 150, 142 degrees centigrade, and they survive out there. They have mechanisms to hibernate. Being a single cellular organism, I mean, the type of gene expression which is being shown by these weird organisms is really interesting to know. So that's why these are basically metagenomes, which are outside or show certain different type of characteristics and belong to organisms which are unicellular organisms, unicellular in nature. So I had a few, thank you again for unmuting yourself. And we have some other, so yeah, partly right, Velu, as I said. And so yeah, I mean, why to study metagenomics was not the question. I was trying to tell you what is microbiome. Now, metagenomics is to study it, and then microorganisms grow in communities and in a way initiate growth of its partners. Yeah, that's also a partial question, but I think I have explained enough. Let's go forward and try to get back to the presentation and go around with it. So microbiomes lives in communities within the body. Microbiomes lives in community within the ecological niche. And earlier, microbiomes has been studied, but microbiomes are very difficult to culture in a laboratory because the kind of conditions they live in, it's very difficult to culture them as a community in a petri dish or something like that. So metagenomics has given that technology, has given that ability to us to take the soil sample directly from the ecological, from somewhere, and kind of sequence it by using special type of sequencing machines, which is the gift of these high throughput sequencing methods. And so as a result of that, you can find many species which are not yet known by the human beings. And therefore, as I said, that it needs no need to isolate them, and also lab cultivation. So when we have these, metagenomes, what do we really like to look at it? What do we really want to find out of that? What we want to find is alpha diversity and beta diversity. These two are the basic things that we would like to know, because we know about the classification, the taxonomic classification of the bacteria till known. I mean, there are maybe possibilities of getting new taxas included into the entire phylloclads or into the phylogeny. So that's why these alpha diversity and beta diversity is important for now. Alpha diversity is something which is within the sample. So you collect a sample from a specific place and try to classify, try to sequence those organisms, and then try to find in one sample from a very specific place what are the different type of species that are there. So as you can see, this particular bar, it looks like a bar graph. So we basically study the abundance of this bacteria. If you remember, the RNA-seq pipeline, we were trying to note in the table of expressions, we were trying to understand some quantifiable things, some RNA expression. So these are table of expressions. Here, the number of reads, we basically try to quantify the number of reads. Here, the entire thing is very different. We try to figure out the abundance. So the number So the value of this sort of experiment is not in decimals. They are mostly in real numbers. They are kind of whole numbers without any decimal part. So as you can see, the alpha diversity, what we take, for example, in this graph, you can see there are 1, 2, 3, 4, 5 phyla of bacteria which are present. Bacteroidetes, Firmicutes, Proteobacteria, Tenericutes, and Verrucum microbia. And this is alpha diversity within the sample, and this is called beta diversity, which is between the samples. So samples collected from, let's say, North England, or samples collected from Russia, or there is some specific place in Russia, maybe have. So there are some experiments, as far as I remember, has been going on in Chernobyl for a long time to really understand the change in the microbial population after this nuclear fallout occurred. So they found very interesting things. They found much the microbial population has changed its nature, and also has gone through interesting changes, which are some adaptive changes, and by virtue of certain genes, maybe of those copies of genes are also present in humans. So it is interesting. No, they have a biological significance and also a scientifically important things. And what we really look at when we decide alpha diversity and beta diversity, we look at the variable regions. So hypervariable regions, we call it hypervariable regions, like V1, V2, V3, V4, V5, V6, V7, V8, and V9. Now these are specific sequence sets which are kind of conserved, highly conserved within species. For a particular species, the hypervariable region is highly conserved, and they are highly variable between the species. So you can understand why do we really take into considerations at the factor of differentiation we take in hypervariable regions as a consideration. So this is what we look at, and to understand alpha diversity and beta diversity, what the elements that kind of we require. We will go into that a few moments time. I would really want to take a pause here, and I would ask you that what do you think of this graph? What is your idea about the graph? You can see many regions of the human body: gastrointestinal region, urinogenital region, skin, and also oral region, and nasal region as a whole. Can you tell me what this graph is all about? I mean, what is your idea about it? What do you think about this idea? People can unmute their microphones and also tell me. I would be happy to listen to your voice. Different microbiomes. Well, yes, specific species of microbiomes are enriched in each region. From oral, nasal, urinary. Yeah, we all know that oral, nasal, genital. So yes, partially correct. So you people are like sequence reads. We have to align them together and then make it into a complete sequence. So different microbiomes, species, distributions of microbiomes in different parts of our body. Yes, of course. That's the first idea that you can get. It's a PCA plot showing clustering of microbes, abundance and diversity. Yeah, it's a PCA plot. It's some sort of a PCA plot. It's not a PCA plot. So this is something which is different than PCA plot. Each microbe has their own sequence, which can be clustered differently. Okay, Prakash. So all of you are kind of partially right. All of you are kind of 60 to 70% right. So what I'm trying to, so if you collect all the information together, is that this is the distribution. This data has been taken from American Gut Project. So American Gut Project is a citizen science project which is being led by Dr. Rob Knight. Dr. Rob Knight has been, probably you have seen his video in Introduction to Metagenomics, Introduction to Bioinformatics course in our educational platform. So why this plot was done? The plot was done, the data from the American Gut Project was put into this, into this plot. It clearly says that the bacteria belonging to the gastrointestinal region is different from the bacteria that is present in the urinogenital region, and which is different from the nasal region and also from the skin region. As you can see, the oral region microbiome is totally segregated to a different place. So think about, the oral microbiomes getting into the gastrointestinal region. So, this is how it happens. This is how infection occurs. I mean, unknown infections within the body occurs. So when some microbiome in the gastrointestinal region infect or by any chance come into your oral region, or from the urinary region comes to the oral region, it can cause infection because it's a foreign place for him to reside. And thereby they would kind of elicit certain immunological responses which would be detrimental for your body. So we need to understand that what sort of bacteria are there. So this is the product of, this is the result of one of the significant study as a part of American Gut Project. You can go to their website and find out. And also you can go back to the introduction to bioinformatics slide. I think I have talked a little bit about that project as a whole. Now, I was telling you about, we try to calculate the abundance of the microbiota. And I was telling you about alpha diversity and beta diversity. These, you separate out the samples. How do you separate out the samples? What do you really want to find it? What do you really want to understand? For example, as you can see, there are 3 samples out there: sample A, and sample C. Sample A have 4 species out here: Pseudomonas aeruginosa, Pseudomonas argentinensis, Pseudomonas flavescens, and Escherichia coli. Sample B have Pseudomonas aeruginosa, Pseudomonas argentinensis, and sample C have Pseudomonas aeruginosa only. So sample C might have some, if you go back to the beta diversity, alpha diversity slide, if you really sample, the beta diversity slide, the graph, this is somewhat like hierarchical clustering, which is also a machine learning principle we will be learning about in the due course of time as a part of this course. So it is more like a beta diversity. So, this is something, if you look at the beta diversity, then we would be understanding what are the components of the sample A versus the component of the sample B versus the component of the sample C. So observed species. So this is, the primary separation before actually when the metagenomic data come over is we want to separate out the sample. So sample A has 4 species, and sample B have 2 species, and sample C have 1 species. So the conclusion from this simple study, so this is what is done actually, when it comes to metagenomic profiling of samples. We try to understand which of the samples have more abundance, more variability. So that is also very important to understand, because that's a course, in the course of study, you try to decipher it in simple steps. So we can conclude from this table that the sample A is more diverse than sample B, which is again more diverse than sample C. So for alpha diversity and beta diversity, the elements what we require there are 2 basically. One of them is ribosomal RNA or rRNA, and another one of them is OTU or operational taxonomic unit. These are these are keywords. Operational taxonomic unit is a what should I say? It's a pointer. It's a profile of a particular species. I'm forgetting the exact word for it, but I think I will be telling you in a little bit of time. And using ribosomal RNA and OTUs, we try to decipher, try to get the alpha diversity and also the beta diversity. So ribosomal RNA are of basically 2 types: prokaryotic and eukaryotic. Prokaryotic 70S ribosome. I think all of you know about the S out here. And eukaryotic ribosome is 80S ribosome. It has 2 subunits, 50S subunits, 30S subunit. So 50S stands for the type of protein that is being used to code 50S RNAs, 23S and 5S rRNAs, 34 proteins. So, and this is the larger subunit, and this is a smaller subunit, and eukaryotic have 60S and 40S subunit. Now, can you tell me why we use ribosomal RNA as one of our chief factors of classification in bacteria. I mean, so ribosome, as you know, is a part of the protein machinery, right? And why do you think the protein machinery or the ribosomes are used as an element of classification? Yeah, Swati, you were right, somewhat. It is universal, as organism cannot live without them. Yeah, yes, molecular chronometer. Uh, that's pretty heavy keyword. Can you just explain, Arvind? H. pylori, acidity causing highly conserved. Yes, very much true, highly conserved. And the protein machinery is the basic, basic molecular biology machinery that are there in almost all organisms across the globe. Because that's the most stable part. That's the any difference in them would reflect, yes, species. Yeah, I would not say species divergence. I would say gene expression would be different. Species divergence is something very big. So understand the rate of evolution. Yes, of course, you can understand the rate of evolution, of course. But again, these are kind of, if I answer you one sentence, the ribosome is a part of the protein machinery, which is inherent to all organisms. That's why that's a basic unit of mechanism which you can find, like the DNA. If you really look at the central dogma, DNA codes RNA, RNA codes protein, and one of those elements of that machinery would be the ideal one to really figure out what sort of any changes produced in that machinery would really be reflected as a part of the life cycle of that organism. That is why 16S gets classified. Exactly, Swati. Thank you very much. So I'm pretty happy to see that many of you are kind of experts. So needless to say, but it's always good to go to the basics to really understand stuff. Now, yes, let us go back to the presentation. Okay, so the next is that, so these are the rRNAs, the ribosomal RNAs. As you can see that these RNA belongs to Thermus thermophilus, the small subunit of the RNA, and the Escherichia coli. Now these are different. That's why we know that from which organism it has come from. So you can go to this link. There are interesting images, ribosome image, tRNA, rRNA images out there. You can use them for your publication, or you can use them for your understanding as well. So going forward, so the procedure that we use, is called metagenomics, and metagenomics is of typically 4 types: 16S rRNA amplicon sequencing, 16S full-length sequencing, 16S rRNA shotgun sequencing, and full metagenomic shotgun sequencing. So it are connected to cost and also flexible read lengths. And the machine that out here, what you can see is MiSeq machine, MiSeq DNA sequencer produced by Illumina. And these are used for sequencing all those metagenomes collected from the natural sample. Universal PCRs are used, amplified. These are amplified. The amplification only takes place in the hypervariable region, because we know that across species, across different within a particular particular within the same species, same set of species, they're highly conserved but differently variable in different species. That's why we use the we have to amplify all those hypervariable regions. And we use the 15 kilobases, the 1500 bases, which are typically the length of those hypervariable region. And this is, so we sequence each of these. Instead of getting the whole sequence done of the entire RNA of the organism, we try to specifically sequence only those hypervariable regions. And therefore we try to understand the alpha diversity and the beta diversity. So full 16S rRNA is approximately 1500 base pairs long, 1 kilobytes long. That's why if you go to the this graph, you can see that that particular region has been amplified, and we take and sequence that particular region to go on to our next level of analysis. Okay, so among the lot of these variable regions, we are interested in the hypervariable region 4, and hypervariable region 4 is the one which we are interested in right now. And these are I would say that these are the sequence is done from the both ends as a paired end. So I think all of you have some clear idea about what we really need the pair end for. So, but if I may ask you, why do you really need the pair end? So, because the reason why I ask you this question, because all of you has completed the transcriptomics part. So, you by this time you would be able to understand. Okay, Velu has a question. How is the full length and 16S amplicon different? So, full length sequence is entire RNA, everything. Not considering the individual hypervariable regions, is the entire rRNA of that. So 16S rRNA, 16S RNA is the specific hypervariable region that we are interested in. So that's called 16S RNA amplicon. So can you tell me why do we really need always paired end as our study? Thank you, Velu. You're most welcome. Cost matters, yeah, of course. Cost will matter. At the end of the day, cost matters, but important. So complete genome costs a lot. So small genomes cost less. But I mean, to amplify both strand plus and minus strand. Yes, but yes, of course, paired end is that one only. But why? Why do we really need? Can't we do with single end? Can we do it single end? Differences incorporated by tuck may mislead. Kind of true, yes. But I'm looking for a specific keyword which is connected to statistics. Sequencing from both ends gives more efficiency. Yeah, I would kind of replace the word efficient by confidence. Sequencing from both ends gives more confidence. So when you read the sequence from one end to the other, and also from 3' to the 5' and from 5' to the 3', so it will give you more confidence. You would say that okay, at the particular position you have found bases from both the ends. So it's like giving you a higher confidence to your cover, to your sequencing. That's why we kind of use paired and read. Okay, thank you very much.
Yeah, I have a question.
Yeah, sure. Can you introduce yourself?
Yeah, my question is from wet lab. If our cell line is contaminated with bacterial contamination in our cell line, so what we should use the variable region like the V1, V2, V3, 16 for 16S RNA primer to check the bacterial contamination by PCR.
Okay, so you want to know
that variable reason we should check for that. Yeah, we can take any variable region like V1, V2, V3 up to V9.
You can take anything, but here we are interested in the V4 as an example. You can of course take, I think, but again, it depends upon the type of species.
No. We have no idea about which type of bacterial contamination in our cell line.
Understood.
Yeah. How can we decide which type of bacterial contamination? If we can see in our cell line, that is the moving particle. According to that, we should do 16S RNA primer for PCR, and we can check our bacterial contamination. For that, I am asking which reason we should take for.
Yeah, so there is an interesting project, I would say, we have educational platform which is called SF9. So that was connected to the exact thing that you were talking about, contamination in the cell line. So the cell line was contaminated, and therefore you can use the pipeline is one of the pipelines that we have which is the normal metagenomics pipeline. You don't have to use, you have to take the entire sequence in that case.
No. If in PCR, if we got any band from any of the region like V1 to V9, then there is a contamination. There is a chances of bacterial contamination in that particular cell line, and we can there is bacterial contamination. But
of course,
my question is it specific for any bacteria, or it is
so you are trying to ask me that which variable region would you use to really separate out the your question, if I can understand correctly.
What V1, V2, to up to V9, what reason I should take to check the bacterial contamination?
Yeah, so there is no specific, I cannot say it's written on the wall that which one you should be using specifically, V1 up till V9. So any of them can be used. As far as I know, that anyone because as far as the knowledge goes right now, any one of the variable region can be used.
We should take one reason or more than one reason.
It is better to use one region and then again go into the other region and really try to figure out, because we have not been able to really, because we have some knowledge about what type of variable region is specific to a particular type of bacteria. I can refer you a paper in a reference publication which might help you to clearly answer your question. Okay, is that all right?
Okay, thank you.
Thank you very much. Thank you very much, Jyoti, for unmuting yourself. So yes, we are looking at, from the figure, I would say that region from V5 to V9 as they appear to contain most SNPs. Makes sense? Makes sense? Yeah, Velu have answered it pretty clearly. So V5 and V9. So, but then V4 is not there. So why are we interested in V4? So I think you have a basis. Maybe you have to be supported with certain from the theory, from the mapping itself, it is apparent single nucleotide polymorphism, but I don't know whether we can really take SNPs from bacteria. Do we take SNPs from bacteria? I don't know. I have no idea. I think SNPs are specific to eukaryotes. So anyway, I think we should move on, but I would keep that in mind, Velu. I would really like to go back to my desk to study about, but if your theory is correct, that from V5 to V9 is the region that can be used for classifying the bacteria, which means V4 is like something which should not be used. Maybe more V2 and V3 used, but V4, people are using a lot of V4 to understand, to classify the metagenomic samples from the American Gut Project. So I would let me get back to you sometime to really answer your question in a very definitive way. But nice answer, nice effort, I would say. Okay, so going forward, so what do we have after the hypervariable region is formed? We have something called operational taxonomic unit. So operational taxonomic unit are those which are proxy species. They represent the species. We kind of sequence of the specific hypervariable region, and OTUs are called the representative of that particular species, and they kind of we align them properly, and and try to calculate the abundance of the OTUs in a particular sample. So each OTU belongs to a different set of bacteria. So that is why we call it, each OTU is a proxy species of that particular sample, within that particular sample, which represents that particular species, because we don't have the entire RNA, we have only the variable regions with us. So that particular variable region is the representative of that species. So Jyoti, it answers your question partially. Maybe you have to look for all the hypervariable regions starting from V1 to V9, and one of each of those steps will really help you to establish the fact that which bacteria is present in your cell line contamination. But I would get back to you with some more definitive answer. But as logic could tell me, this is the kind of answer that I came up with. but I hope this helps. So let's see, I would get back to you for sure. So, and then what we try to do, we try to classify these samples into hierarchical clustering. So this is the phylogenetic tree we have built out here. If you can hold on for a second, I have to just plug in my computer to the power cord. I think we are kind of. Okay, so sorry for the interruption. So technical glitch, I guess. So by the OTUs, we try to build a consensus set of sequences, and each sequence in that consensus pattern represents a species. We try to classify that into some sort of, in the entire phylogenetic tree, we try to place those proxy species in specific locations so that we know what are the species of microbiome that are present in our sample. Now, why is it important to classify that into phylum? Because the type of species residing in the skin would be different than the type of species residing in the oral cavity, and also which will be different from stomach. So each one of them is different. So see, the percentages of firmicutes in vagina is higher than the percentage of firmicutes in the skin. So therefore, we know that microbiome is considered as an organ by itself. So we would be able to know that what is the health of that organism. That organism really dictates the entire metabolic principles within our body. That is why all those metabolic processes within our body. That's why we need to know that what kind of bacteria and what is the health of those bacteria within the body, and thereby we would be able to say that what is the level of infection caused by that bacteria. And we all know about what is biological classification: species, genus, family, order, class, phylum, and kingdom. And I mean, more number of species as we go up, it's a kind of a pyramid shaped structure. And this is an interesting thing that we also like to share. The typical database that we use to ratify our results in the pipeline is Silva, because Silva is the database which I think, Jyoti, if you're working with microbiome, I think you would be using Silva and Green Genes. But of them, Silva is the most updated one because it is maintained properly and you get the right kind of information. We also use Silva and Green Genes to ratify our results. And now let us go to the pipeline. Let us go to the server and figure out what all we have there as a part of the pipeline. Okay, so yes, let us go to the server. So I guess all of you have been able to log on to the server by using your credentials shared to you. I have already logged on. So I will take a moment here to show you, to allow you to go back to your server and log in. So once you log on, you can see, if you click on this person on the corner, you can see your login name. So see, I'm logged on. That's why I can see my name out here. Once you have logged on, go down in the NGS bar in the NGS section, and you can see something called amplicon 16S and 18S. So 16S belongs to bacteria, 18S belongs to those eukaryotes which are residing within our body, 18S. So 18S sequencing is 18S sequencing is mostly used when it is coming to gut bacteria, etc. So have you logged on? I guess. So, so this is the pipeline that I wanted to show you. This is a demo pipeline. Okay. So demo pipeline helps you to understand how the pipeline is being built, and also tells you what can you expect of the pipeline at the end of the day. So the AI guided platform, I think all of you are kind of aware of it. So let's click start. Now this is the input file. What are the input files we have? We have several input files. The most important input file that we would be interested in is links.svl. Now what does it have? Let's see. This is the type of file that you would be like to look at. Okay, it's being saved. So let's see it in the folder and then open it via some notepad. Okay, we can see something here. Can you tell me what is it? I think you people are kind of now are in the level that you can say what these links mean. We have an answer in the chat. Raw reads file. Yes, Velu, raw read file, definitely. But what sort? Yes, FASTQ file, correct. Are these paired end or single end? So Arvind has asked the question, can we call the identified entities as species? Yes, that's the whole idea. That is the whole idea, that you can that is the whole idea, to identify, to classify them into different species. Yeah, paired end 1 and 2. Yeah, so 1 and 2 both, so paired end. So that's why two sets of FASTQ file. One is this, and the same accession number by _2. So these are paired end reads in the server. So as I'm telling you that the FASTQ file are very large file. These are kind of big data of the file. Metagenomes is the first organism in the entire world of big data, where on which big data bioinformatics, this is the first biological big data came into existence from the domain of the microbiome. So because they have huge, I mean, the number of, the amount of information which came out from the high throughput sequencing is huge. Okay, so yes, so this is the paired end file, and this is the soft link of the file that are located in our server. And what you have to do is, you can just, we are kind of taking the hassles from you to build the to download the FASTQ file and upload it out there in the server. We have already uploaded that in the server, and we are just giving you a link. Remember that when the time comes for you to do the project, we would expect you to give us the links of those FASTQ files. If you're working on RNA sequencing, if you're working on genomics project or transcriptomics project or metagenomics project, or any sort of other project that you want to work which would use our pipeline, we would be uploading the data for you in the server, in the backend, the project which you are going to do at the end of this course. So the data sources has to come from you, or else we can also help you to find the data sources. But then the biological question, we would expect that the biological question has to be defined by you guys. So that's why these concepts are important, that's why these concepts are very important to know, that what are these type of files, because and where do these files really come from. Okay, so let us go back to the pipeline. So this is the input file, right? And we have some reference URL list, don't bother about it. And we have something called chow and HFD. These are specific data set from mice which are fed normally, and also high fat diet. We'll be looking at this type of data as a part of the project in our next class. Now let's just run the pipeline and see what does it produce. Okay, so we are running today something called data pipeline. So data pipeline is actually brings out the OTUs, the operational taxonomic units, from the metagenomic sequences. So this is the data pipeline. The data pipeline is a proprietary algorithm developed in Tauber, which concerns, which connects the QC filtering, replication count, pair merge, cluster consensus. This is where we build the OTUs. So before building the OTUs, there are several steps which goes into it. And then removing chimeras, because chimeras are important, because chimeras are those sequences which are kind of mix of a number of bacteria, more than two, more than 2 bacteria. We do not know which particular species does it belong to. So we have to remove all those artifacts. We have to, that is why it is something which is very important, to really identify new species of bacteria in your sample. And then assign the taxon. This is the annotation. And thereby we use something called phyloseq to build the alpha diversity. So we build the alpha diversity by using this pipeline. So this is the place where we go. So let us run the pipeline. So QC filtering and these small educational pop-ups will give you the idea that what is this data pipeline all about, and what actually, forward reads and reverse reads, which are good quality reads, which are bad quality reads. So if data pipeline will also tell you that whether your reads that come up from your experiments are really good reads or bad reads. This is very important. So this can be also a way, Jyoti, to find out the contamination in your sample. Okay, so there's a publication associated for that. I think you want to go back to that and read the publication. That will give you more idea. And this is pair merge. And then going forward to do the OTUs. So see, demo pipelines are basically, you don't have to bother about anything about the parameters that go into building the pipeline. So you know what are the input files and what are the output files. So we have the ape as a phyloseq, and we kind of finish it up, and then we build it, and we end. Okay, let us see what we have here. We have something here. See, we have so many things. We have an abundance table I'm talking about. Let us see what this abundance table contains. Okay, it's been downloaded. It's been there in the folder. And this is something which I would like to open with a spreadsheet program. I'm using Linux, so I'll be using something called LibreOffice. Okay, now we have some interesting thing. What do we have here? I think all of you are. See what I was telling you. So OTU1, these are number of OTUs or operational taxonomic units that has been found out from this sample. So there are 305 OTUs. So it is possible that there are 305 different types of species of bacteria present in this sample. And each sample is BKF01, BKF02, BKF03, BKF13, BKF14, and BKF15. Okay, so within the sample, see, I was telling you that the abundance table would contain whole numbers. See, this is the number of OTUs in this particular sample. We have not assigned the taxon. So the taxon, we have already assigned the taxon. Sorry, we are going to see the taxonomic. Let's look at the phyloseq plot. So that's the PDF which is being produced. Let's see how it looks. Okay, we have something interesting here. Yeah, we have something. We have some alpha diversity. Let's fit page. Okay, great. See, we have so many types of abundance. This is the abundance plot that we would really like to look at. And so many species are there. See, and some unknown species are also there. So that's also important. And this is a heat map that is being produced according to the OTU abundance within the sample, between different samples. And this is the beta diversity, that abundance of the samples across different species based on the OTUs. And this is something, some plot which you have looked at in the American Gut Project sample. See, we would discuss more about this pipeline, the results of this pipeline, when we go into, I'm going to describe two projects to you One is, one is an American gut project, and another one of these, a gut-brain axis project, which we would like to tell you, and the findings of it. What did we really yield out of that? So these plots are being generated out here. See, so see the separation of the species, the alpha diversity measure, and these is the similar to the PCA plot, the principal component analysis plot. So we use something else, that PCA. We use something called NMDS1 and NMDS2. This is similar to PCA, but again, this is different than PCA. Okay, now let us go back. So this is the pipeline we talked about, and this is the abundance table that we talked about. You can really find that what is the distribution of these abundance by putting them into a plot. Maybe let's say I can plot it up. So this is the abundance that you would really find out using the spreadsheet program. You can do different visuals of these particular abundance and for your own understanding. So different, let's say if you kind of you want to understand these 4 OTUs across all the sample, then maybe you can plot it like this, right? Okay, so the annotation is not very clear here because I have not given an annotation. So each of these, in sample 1, we have so many, we have 4. So these are, sorry, this is OTU1 and this is the number of samples. So OTU1 is present at this level in sample 1, BKF1, BKF01. This is in BKF02, this is in BKF03, this is in BKF013, this is BKF14, and this is BKF15. So you can find the comparative abundance of each OTUs in across the samples in your result of the data to pipeline data. Right, so this is the demo pipeline I talked about. I think the best thing would be to go back and run the demo pipeline and try to understand that, and also go through these 2 educational courses. This covers all the concepts I kind of touched upon today itself, the introduction to metagenomics and metagenomics 1, and this talks more about in fact, the introduction to metagenomics is the best place to start from, to understand why really do we really need to look at the metagenomes. That is very important. And why do we really need to classify them into taxonomic classes and try to understand their infections, try to understand their biology, and also try to understand what sort of pathways do they really belong to. This is one publication that I would really like you to read. Why OTUs are important. Identify threshold for 16S ribosomal RNA OTUs. Maybe this is, you have already come across it, or maybe you have already been. Those of you don't know, it would be a good place to visit. So I think we are kind of out of time today, and I can take 1 or 2 questions, and then I would like to tell you again about the feedback forms, the pre-assessment form which has been shared to you, and also the transcriptomics form that has been shared to you. So we would really like to know your needs. For example, the question that Jyoti asked, that how that does she really separate out those samples, the cell line contaminations. So Jyoti, the SF9 project we have in the educational platform, I would kind of want to talk to you regarding that, and probably we can help you out to build the right kind of pipeline in the server to separate out the cell lines, and which hypervariable region would be really important for you to find out the contamination. The next class is on September 24th, which is closed, and we'll be talking about metagenomics 1. We want to talk about the current research areas, the hot topics of metagenomics that which we are dealing with, the logical steps of the analysis pipeline. I mean, we already showed you the data to pipeline. Maybe the next day I would really like to build a particular pipeline using some sample data set, the methods of interpretation results, and visualization as a whole. So I would like to give the stage to Dr. Mohit Majumdar. So there is something which is a program which is coming up as a part of our the online program, which is called Biomedical Data Simplified, which has already started yesterday. Maybe some of you visited that. Maybe Om Prakash visited it, and many of the other guys who are kind of interested to know about the applications of data science in a more greater way, like exploratory data analysis, or let's say inferential statistics and descriptive statistics. This is a perfect place to be. So I would like to give the stage to Mohit, and if he can say a few words about it. I have a question. Sorry, Mohit, just hold on for a second. I have been accessing the education portal, but my score is not being reflected. Arvind, we will look into it. Definitely, we have got these complaints from many people. So I think there's a problem. There's some bugs in our server. We have already identified the server. I think Apar is here. Apar, could you please take care of this stuff? Ask Joshua to take care of it and solve the problem. Yeah, Arvind, we will take care of it. Don't worry. So yeah, so I would like to, welcome, most welcome, Arvind. So yes, I would like to give the stage to Mohit. Mohit, are you there?
Yeah, thanks, Pratim. So yeah, yesterday we started with the introductory session regarding the biomedical data simplified. So this is a data science course which talks a lot about biomedical data. So, but we'll be discussing machine learning and the approaches in more details. We'll also go through those things in this course, but we will have an overview. But the other course that which is started, and anyone is also invited to join our program, because we just had the introductory session, and as you know, the videos are always available to go back. So it's a great opportunity to to enhance your learning using the techniques that are ongoing. We'll be talking about some industry examples as well. So all those people who thinks that this will help them in their best way in terms of their academic or research or industry research, so they are most welcome to join us. So we are also giving a lot of discounts. So let us know. Thank you, Pratik.
Yeah, thank you, Mohit, for kind of elaborating on the fact. And we have a question here in the chat. Is this some comics? Yeah, it's not comics. Yeah, it is omics data science. Yes, absolutely, it is omics data science. It is multi-omics data science, to be very specific. And in the data science part, we would be covering a lot about intricate statistics and all those exploratory statistics, and we'll be using how can we join. Nidhi Shah asks, how can you join? So Nidhi, you can, Apar, can you help Nidhi to let her join in the course? So there is something, Apar, are you there? Okay, Nidhi, so I don't think Apar is, I was here. So let me share my email with you, pratim@pine.bio. And you can email us at mohit@pine.bio. You can email us, Nidhi, just send us an email and we will sending you all the details for you to join the course. Thank you, Nidhi. Okay, so this is a very interesting course, and where we so coming back, we would also be teaching you, the specific type of analysis which you can do via programming. So we will be using RStudio and the R scripts primarily, which is some of the pipelines in our server generate generate R scripts. So how to use those R scripts? How to use those R scripts to really classify your data in a more better way, visualize your data in a more better way, and really have a visual classification or a visual understanding about data. Because big data, as you know, or if you don't know, I mean, let me tell you that big data is all about visualizing stuff. So there's a lot of effort going on across the globe to visualize the data in a finite way. So reduce the dimension of that data and come up with procedures to to visualize it in a proper way so that you can have a visual separation of the data. The graph that I showed you on the, on the American Gut Project, if you can see distinctly that, okay, urogenital separates from the gastrointestinal separates from the oral and the skin. So this gives you a very good understanding. So you know that what samples you would really like to look at. Maybe you can really understand that what are the converging regions of between all those samples, and maybe like to study all those essential consensus sequences or consensus species which are abundant in all those places where which are common species across all those locations. So there are several scientific questions you might ask, and there are several translational aspects which you might be interested in. So that is how we will be using all those materials and methods. So I think let us stop today, and then kind of I think we have overshoot the session. Thank you for attending, and we are looking forward for you to join the class. In the meantime, please fill up your forms, and also go to the educational platform and do the necessary courses.